everyone, welcome to A Plus Biology class by Esmer and here, and today we are going to solve October-November 4-2 paper of 2023. So October-November 2023 paper 1 hour 15 minutes is the time, and let's start. The question number 1 is red blood cells are specialized cells that transport oxygen. State the substance in RBC that transport oxygen. So what will be the name of the substance which is present in the RBC? The name of that substance is hemoglobin. Okay, then state the name of the component of the blood that promote blood clotting. That component is platelet. Then next part, they have given the RBC which is immersed in the different concentration. They have shown the initial diameter of all the RBC. Here they have placed the RBC and they measure the diameter 7.5, and the concentration in which they have placed in the 0.4, 0.8, 0.9 and 1.8 different concentration of salt solution is taken. In the first one, cell burst. In the second one, the cell increase in diameter, same in diameter, decrease in diameter. So this is the situation. Calculate the percentage increase in the mean diameter of RBC, and that were immersed in 0.8. So 0.8 is this one. It's this concentration was the initial and this concentration was the final. And if we calculate the percentage increase, the formula is always final minus initial divided by initial in 200. So let's put the values. So that will be 8.2 minus 7.5 divided by 7.5 in 200. Okay, so 9 point something, that will be the answer. Explain the result that RBC that were immersed in 1.8% salt solution. So if we look at 1.8% salt solution, its diameter decrease. So when you explain this, it means whenever it placed in that solution in which there is more salt outside, so water move out by osmosis, that's why its diameter decrease. So the reason is that explain mean water move out by osmosis, and how the water is moving out from high water potential to low water potential. Okay, and through membranes, through RBC partially permeable membrane. Clear? So this is a way how this 3 marks question will be. You can achieve the 3 marks. State why there is no change in the mean diameter of RBC immersed in 0.9. So if the water potential here and inside the RBC is same, in movement and out movement is same. So this type of condition is called isotonic. Why? Because the net movement is same. The net movement in and net movement of water in and out of the cell will be same. Next is state why red blood cells burst when immersed in pure water. Yes, what happened in the pure water? So this is RBC. Why it get burst when the water keep on entering? The reason is that there is no cell wall, because more water enter and it will not withstand high pressure, due to which the cell will burst. State the two uses of water in plant. First, it is the main reactant of photosynthesis, because the process of photosynthesis need water, and also it acts as a solvent because all the materials dissolve like ions, like sucrose, they dissolve in it and then it moves. And you can also mention it maintains turgidity of the cell. Here they have shown a water lily plant which is a hydrophyte. Hydrophyte means it is adapted to live in the water, and describe what is meant by term adaptive features. So you can see they can float on the water surface. It means it have some adaptive features. But what is the adaptive features? The adaptive features mean it is any inherited it is inherited structural features inherited structural features, and any that inherited structural features which help to increase the chances of survival. So increase the chances of survival. If it is increasing the chances of survival, it means that is the structural feature which is helping to adapt in that environment. So here they have shown the inner structure of that leaf. The A will be, you can see the A is cylindrical, palisade mesophyll, then B is spongy mesophyll, and then C is air spaces. And next part is explain why the sea adapt the hydrophyte to its environment. Because you know that the adaptation of this. So if there is more air spaces, so how it adapt? It can float easily on the surface of water. So due to the air spaces, it provide buoyancy, or you can say it float easily on the surface of water. Then what happen? Then it can capture more sunlight. So capture more light mean it can go for more photosynthesis. So that's why how the hydrophytes have more air spaces. Here they have shown the mean number of stomata in the tomato, upper epidermis, lower epidermis, and look at the water lily, the more stomata on the upper epidermis, and look at the tomato plant if having more stomata on the lower epidermis. Let's compare these two. You can give the reference of the data. You can write the similarity that both have stomata, both contain stomata, and you will mention that water lily have 475 on upper surface. So you can give the reference of the data, you can quote the data. Upper epidermis, even you can mention about the uh, tomato, but the question is compare. You have just this comparison, 2 marks for comparison, and then explain why this is so, why the tomato have uh, more number of stomata on the lower epidermis. So if we look at the tomato plant, it's exposed to sunlight. Okay, so tomato exposed to sunlight. So when it is exposed to light, it means to reduce transpiration. To reduce transpiration, it have less stomata on the upper surface, more stomata on the lower surface to reduce transpiration. But if we look at the water lily, it have more stomata on the upper surface, and the reason is that to for because for the gas exchange, it's needed for gas exchange, and because photosynthesis occur, gas exchange is needed, so stuff, and it is the water plant. So that's why the lower surface have no stomata, or because stomata is not needed in the lower surface. State the name of the cell that control the opening. This type of cell that control the opening, that is called guard cell. Next question is that they have shown the synapse with two different situations. One is a normal person, and the second one with uh, disease, Parkinson disease. So the healthy person synapse is shown this one, and the Parkinson patient synapse is this one. Easily you can differentiate between them. Look at the neurotransmitter containing vesicle, look at the neurotransmitter release, look at how much is binding with the receptor proteins. Identified label X. So label X will be vesicle, or you can say vesicle membrane, because they are not pointing towards the neurotransmitter. So vesicle or vesicle membrane, and Y will be synaptic cleft, or you can say synaptic gap. So next is using the information, suggest and explain the effect of Parkinson disease on a person's movement. So if we look at the question, this is a 5-mark question, and Parkinson disease with the neuron, the answer is in the diagram. You can see easily how it is different. The person who is having Parkinson, you can see that is that with Parkinson there are less vesicles, less neurotransmitter release. If there is less neurotransmitter release, then what will happen? So look at this diagram. Less binding of neurotransmitter with the receptor proteins. So less binding with neurotransmitter, less binding with receptor protein. So neurotransmitter less binding with receptor protein. If there is a less binding with the receptor protein, then it's a slow response, mean passage of nerve impulse will slow down. So basically when you write the suggestions, you write that it's slow reflexes or slow response. And explanation is that point which you have mentioned all these points in the explanation. So this is a reason why that person have slow response or slow reflexes. Describe two ways the nervous system differ from the hormones. So when we write the differences, the nervous is always faster and the hormonal is slower, and the nervous system in the passage of impulse through neurons, because neurons involved, and here the hormones released or secreted by glands, and they are the chemicals, while chemicals which is secreted in the blood, while in this case the neurons which carry impulses, neurons carry impulses from one place to another. Okay, then explain the shape of the receptor proteins is determined. Shape of receptor protein, basically this shape is determined by sequence of amino acids, because different proteins are different because of amino acids arrangements, and then this amino acid arrangement is decided by the DNA base sequence, because in the protein synthesis the DNA is copied by mRNA, and then a protein is synthesized according to that sequence. Cell membrane also contain protein carriers, and what is the role of protein carriers in the cell? If there is a membrane and that membrane contains such kind of carrier proteins in their membrane, so it means it is for the movement of molecules in and out of the cell, that movement of specific molecules, and specific molecules like they can be ions, glucose, amino acid, and they can move by active transport from in and out of the cell. So they can pass through the carrier proteins by active transport. Here they have shown the effect of temperature on rate of photosynthesis in one species of the plant. So photosynthesis is enzyme controlled reaction. Remember that this is enzyme controlled reaction in which rate of photosynthesis is shown and you can find optimum temperature. So look at the maximum rate. Where is the maximum rate? That is the 40 where there is a maximum rate. The centigrade unit is mentioned. Explain the result in this figure. Okay, if you say that 20, 25 to 40, the rate of photosynthesis is increasing, then you have to mention the reason why the rate of photosynthesis is increasing here. So rate of photosynthesis is increasing. The reason is that more temperature because you are increasing the temperature. So more temperature means more kinetic energy. So if there is more kinetic energy, it means more collision. More collision, what is the next step? More collision mean more enzyme substrate complex. So more enzyme substrate complex. So this is the reason why the rate of reaction is increasing, but you can mention at 40 it's max mean maximum enzyme substrate complex formed. So look at the situation after 40 why the rate of reaction is decreasing, then you have to mention the reason because this is a high temperature. At high temperature the enzyme denature, if the enzyme denature and the shape of active site disturb at high temperature, then the rate of reaction slow down. Carbon dioxide was supplied in excess at temperature because this is an experiment in which you are just taking the plant disk and you are just checking that how much oxygen is produced by photosynthesis. The CO2 level must be same because the CO2 will not act as a limiting factor because we only want to check the effect of temperature, not CO2. So CO2 is in a high concentration, so CO2 not act as a limiting factor. Right? So this is a reason. It's just why not all the oxygen produced by the leaf is released because some of oxygen it is used for respiration. Otherwise, the remaining oxygen diffuse out. Some of it is used for respiration. Describe the role of chlorophyll in photosynthesis. So chlorophyll is a green pigment. What it do? This green pigment capture light and help in converting light energy into chemical energy. In converting light energy into chemical energy which is stored in the glucose molecule. So this chemical energy is stored in the glucose molecule. So this is a way how chlorophyll helps. Next question, number 5. Describe the two ways in which circulatory system of fish is different. So if you look at the circulatory system of fish have the single circulation while human or the mammals have double circulation. Fish have the single circulation and the second thing is that the fish, the blood passes the heart only once. So there will be in the fish there are two chambers of the heart, and in the fish there is no separation of oxygenated deoxygenated blood. So explain the advantage of double circulation. Double mean blood passes the heart twice. So when the blood passes the heart twice, then actually there is a pressure is also created. Pressure mean high pressure which is needed for the movement of the materials. So high pressure is there maintained, oxygenated, deoxygenated blood is separated, oxygenated blood and deoxygenated, they are separated, high pressure is maintained, okay, they are separated. Okay, so this is the way how the double circulation is important. Now you can see the diagram of the circulatory system of the mammal, and here you can label these parts. If something is labeled hepatic artery, it means its next this B part will be liver, because hepatic artery supply blood to the liver. If they have labeled the renal vein, it means that the D part will be kidney, because kidney is related with the renal vein. Okay, we don't know rest of the parts, but we know that if there is a connection between the two organs of the body, then the X is hepatic portal vein. X is hepatic portal vein. Okay, and the C will be intestine, because from the small intestine the capillaries join to form hepatic portal vein, and then it carry blood to the liver. And A is the lungs. So according to this, let's fill in the blanks. The absorption of product of digestion into the blood. Absorption of product of digestion. Absorption is done in the C, right? Which is the intestine, small intestine. Excretion of carbon dioxide from the body. That is A. Lungs. Carbon dioxide is excreted from the lungs. Formation of urine. That is D, because formation of urine occur in the kidney. And the production of bile, that will be in the liver. The answer is B. Is it clear? Identify the name of the blood vessel X. Already we have mentioned, that is hepatic portal vein. Hepatic portal vein. The role of liver in excretion. Okay, you know that in the liver, all the excessive amino acids break down. So all that excessive amino acids break down in the liver. What's the name of that process? The process name is deamination. Deamination is the process in which urea is formed. So urea is formed, which is excreted by the kidney, right? Next part, last question 6 is state the name of the two hormones released by the ovary. So hormones released by the ovary is estrogen and progesterone. Organ, the antibodies cross the fetus, that is placenta, because placenta is the organ through which antibodies transfer, because placenta is formed here in the uterus lining and with fetus tissues, and there is a connection between both of them because of the placenta. So here they have shown a graph in which the concentration of antibodies in the baby's blood, which is decreasing, and then up to 6 months they stop the breastfeeding, and the antibodies acquired by the baby from mother is mentioned with the straight line, and then the dotted line presenting the antibodies produced by the baby. Let's see what's the question according to this. After the birth, the concentration of antibodies acquired from the mother decrease rapidly to zero. So where is the point where it's rapidly to zero? So this one is the point where it rapidly decreased to zero. Antibodies start being produced by the cell called dash. So the lymphocytes are the cells which produced antibodies, lymphocytes. The total concentration of antibodies in the baby from both sources is dash for 4 months at 4 months. So we have to find this is the concentration of the antibodies here And this is the concentration of the antibodies. We, after the both sources, we need to plus both the values, and then the when you plus both the values, the answer will be 56. The concentration of antibodies acquired from mother and the concentration of antibodies produced by the baby are same. So where is the point when these two concentrations are same? That will be round about 3. The concentration will be same at the 3 months. Describe the benefit of breastfeeding a baby in first 6 months, because nutrients, the exact required amount of nutrients are provided. So exact same amount of nutrients are provided uh, from mother to baby, and temperature is also maintained, no contamination, antibodies are transferred. Okay? So it make a bond between baby and the mother. Two ways other than the breastfeeding the baby can acquire immunity. So how the baby can get immunity? Because the vaccine a vaccine course is completed in the starting 9 months of after the birth. The purpose of vaccine is baby acquired immunity, or the second option is any antigen or pathogen enter any foreign antigen enter the body. If the any antigen enter the body, lymphocytes are activated and it provide uh, immunity. Importance of the shape of antibody, because the antibody is the shape like this, and if this shape is complementary to the shape of antigen, so then it can recognize. It should be the importance of shape. It should be complementary to the shape of antigen. So then what happened? It will recognize it as a non-self. It start inactivating it. It uh, uh, help it to capture and also showing agglutination. Antibodies are proteins, state the chemical elements in all proteins that are also present in carbohydrates, that is carbon, hydrogen and oxygen. Okay, so the whole paper is done. I hope the concepts are clear, and if you like my channel, don't forget to subscribe.